My name is Dana Hain. I met all of you last week, I believe, when you attended the webinar. And I'm joined again by Elise Hickman and Victoria Triana, who you'll see every week of the series. And then we have another special guest tonight, Greg Smith, and Elise will introduce him in just a few minutes. But I wanted to just do a few introductory things before I turn it over to Elise and Greg. So tonight we're going to be talking about experimental models for studying flavored e-cigarettes. And Greg and Elise are going to be telling you about the research that's happening at UNC to study flavored e-cigarettes. And then we're going to wrap up the day with a featured classroom activity. So this is a lesson Elise and I wrote back in 2019, I believe. It's a data interpretation activity. So we're going to do that later today and go into breakout rooms. And that's going to give you the opportunity as well to meet some of the other teachers enrolled in this series. And hopefully tonight we'll have time for discussion as well, but we also have worked time for discussion into next week's session also. So here are the goals for tonight's session. We're going to be providing an overview of 3 experimental models being used to conduct research on the health effects of vaping. And we hope as you learn about this and as you conduct our activity, you'll be considering opportunities for integrating some or a lot of this content into your instruction. Everyone here teaches life science, biology, anatomy, or health. So there's some commonality with the educators on the call tonight. And then we will specifically be examining data from a cell culture study. And you'll be drawing conclusions about the impact of both PG/VG, if you remember what that is from last week, and flavorings on cells of the respiratory immune system. So that's a little bit about where we're going. So now I'm going to turn it over to Elise, who is going to begin sharing some science with you all. Feel free to drop questions you have into the chat, and or you could hold them until the end. So Elise will take time at the end of her session to ask questions, and Elise will be with us the whole session as well. So feel free to come back to that. So it's great to see all of you here. Elise, remember, is a PhD candidate in the lab of Dr. Alona Jaspers. That was the scientist who gave you the talk on flavorings last week as well. So she's going to be talking to you more today about cell culture models and also human clinical trials that they're doing within their lab. So thanks, Elise.
Yeah, thanks, Dana. And I see a question, will slides be shared again at the end of the presentation? And yeah, I'm pretty sure we're planning to share all these slides and also the recordings of the sessions. So with that, we're just going to get started with the first micro talk for today, which is about cell culture models in respiratory toxicology. So last time we talked about some of the questions that scientists have about the health effects of e-cigarettes. And for the purpose of this series, we're specifically interested in the biologic effects of chemicals in e-cigarettes on different cell types, on different cellular functions. What is the mechanism of the effects? What diseases could e-cigarettes potentially cause? But before scientists get around to answering any of these questions, we have to think about what experimental design should we use, what model organism should we use. And even though what we truly care about are the effects of e-cigarettes on humans, it's not always ethical or possible to use humans in experiments. So there are a lot of different things that we can use instead. And they exist on this sort of continuum of how easy they are to work with, maybe how affordable they are to work with. I mean, there are, experiments that you can do with any model that can be really expensive, but in general, this is kind of a gradation of different experimental models, all the way from computer modeling, which doesn't take anything except a computer, to using human subjects in research. And for this talk, we're going to be focused on cell culture. So what is cell culture? It's when we grow individual cells in a plastic dish and they're surrounded by liquid media that provides nutrients and other molecules that encourage their growth and survival. So this is what cell culture dishes look like. You can grow them in lots of different types of dishes, different sizes, depending on what experiment you're interested in or how many cells you have. And then they are typically grown in an incubator, and that environment is supposed to mimic the human body. So it's pretty warm, there's some carbon dioxide, and it's also pretty humid. And this is what those incubators look like. So it does take some specialized lab equipment, also including some biosafety cabinets that keep the cell cultures from getting contaminated and also protect the scientists from any cells that they're growing. but it does offer a lot of flexibility in the ways that you can grow the cells. And there are some advantages and disadvantages of research with cells, like there are with any model organism that you might use. So cells are easy to grow and manipulate. Like we just talked about, you can grow them in all kinds of dishes and different conditions. They're not quite as expensive or complicated as animals, so you don't have to get special approval usually to work with cells. You're not dealing with a whole organism that you have to keep alive. And you can easily treat them with a lot of different chemicals at different concentrations, and you can change up the experimental parameters really easily. But because you are growing them in a dish, you can lose some of the biological relevance of studying a whole organism because they're not in an organ and they're not surrounded by all the other cell types that they would normally be interacting with. Cell culture is also not always representative of the whole population because the cells might only be from one person. So you're really only looking at the effects on the cells from that one person. And even though it can be affordable, depending on the type of cells you're growing, you can be really expensive to buy all the media that you need. And there's always the risk for contamination of your cells or other technically challenging protocols you have to follow depending on what type of cells that you're growing. And for e-cigarettes, we're interested in the respiratory system and respiratory cells. And the respiratory system is responsible not only for gas exchange, like I'm sure you all know, but it can defend the body from inhaled pathogens and pollutants. And all along the respiratory system, there are different cell types that are important in the structure and function of the respiratory system. So up a little bit higher in the nasal passages and then in the trachea and the bronchi, you have thicker epithelial cells that have cilia on them, goblet cells that produce mucus, and these are involved in mucociliary transport where the mucus is beat up and out of the airways. And as you go down into the alveoli, you can see that the cells look quite a bit different. They're a lot smaller and skinnier, and there's a lot more space, and that's where the gas exchange occurs. But altogether, there are about 40 different cell types that make up the lungs. So that's a lot of choices for what cell types you might study if you were going to use cell culture in the respiratory system. In our lab, we're really interested in the respiratory immune system, and we focus on 3 main cell types: ciliated airway epithelial cells, neutrophils, and macrophages. And we're really interested in the immune response because normally if someone's healthy, they have a healthy immune response. You're able to defend yourself from infections, and it's a pretty good place to be. But in either direction that you go from that healthy middle, so if you're immune suppressed, and I think a lot of people are really aware of this term now, especially with COVID and thinking about people who might be susceptible to COVID, they have insufficient host defense and they're not able to defend themselves from infection. You can also have too much immune activation, which can result in inflammation. And sometimes that can cause autoimmune disease where your body is attacking itself. So either way we go from this healthy middle is something that could have implications for health. So we're interested in either way things might go when we study these different cell types. Where do these cells come from if we want to study them in culture? So we use both cell lines, which are human cells. You can also have cell lines of mouse cells or a lot of other organisms that have been modified to continuously and easily grow in a dish. Sometimes these are cancer cells because obviously those grow and replicate really quickly and easily. They can also be healthy cells that have been purposefully changed so that they grow easily. And then we also use primary cells, which are cells that we take from humans, and then we can grow them in a dish for a short period of time, but they don't typically live as long or replicate as much as a cell line. And again, we grow them in these dishes, and sometimes we grow them, they adhere to the bottom of the plastic well, and then they have media on top of them that feeds them. We also grow cells using these special supports called transwells, and that allows, on one side, we can have liquid media to feed them, and on the other side, there's air. And so we can expose them to things that might be in the air, like ozone or e-cigarette aerosol, which allows us to do an exposure that's more relevant to how people would actually be exposed. So the primary cells are really interesting because we do take them from humans. For example, to get macrophages, we can collect those from the alveoli of healthy human volunteers who undergo a bronchoscopy, where a doctor inserts a bronchoscope down your airways, and you're awake for this. I have a lab mate who's done it. And then they wash out the airways with some salty solution, and that can allow you to get the macrophages out of the alveoli. We haven't been able to do this very much recently because of COVID, but it's normally a really great way to get these cells to study. We can also collect neutrophils and other white blood cells like natural killer cells from human blood, because in healthy people, they don't normally have enough of these cells to take them from airway samples. So we can get blood from people, and through centrifuging the blood really fast, separate it out into layers of different cells and then pull out just the layer that we want to use, for example, neutrophils. And then for respiratory epithelial cells, we collect these either from the nose or from the bronchi of human subjects. Sometimes from the bronchi, it's organ donors, and then we grow them in a special type of cell culture well. Again, one of the ones where there's air on one side and media on the other, and the cells become polarized and look a lot like they would look in the body, with cilia on one side and then a more rounded appearance on the other side where they would be touching the body. So when we take these from the nose, it's actually kind of similar to a COVID test. Everyone is, again, really familiar with the nose now because of COVID, but we go in with a little scraper and scrape off some of the cells from the inside of the nose. So once we have the cells, how do we typically set up an experiment? If we're talking about e-cigarettes, we would usually expose the cells either in liquid to a dilution of e-liquid, or we have some setups where we can expose the cells to an e-cigarette aerosol using a chamber. And then we collect the samples and we can analyze them for specific things that we're interested in, like signaling proteins that have been released into the media. We can measure their cellular function, like how well their mitochondria work and how well they're doing glycolysis. For the immune cells, we can also measure phagocytosis. We can take the cells, lyse them, and collect RNA to look at specific RNA transcripts. We can see how many cells are alive or dead after an exposure, and we can measure their responses to bacteria or viruses after that exposure. So there are a lot of different things that we can do with this kind of basic experimental design. And I'm going to show you all this slide because it'll become relevant later, but just thinking about this experimental design with cells, we would always have cells that are just exposed to their growth media and no flavoring chemicals alone, because those serve as our control. And then we might expose another group of cells to a different flavoring chemical, and then a third group of cells to a second flavoring chemical. And we typically repeat this multiple times for each experiment. So we would have this type of experimental setup, and then we would repeat it maybe 3 to 6 more times. And if you think about how many cells might be in the well, it's about 100,000. So that may be kind of hard to visualize, but it's a lot of cells even in these small wells. We wanted to give you a couple examples of an experiment we might do using cell culture. And this was the project that I started with in the lab a few years ago. And I was studying how neutrophils responded when they were exposed to specific flavoring chemicals. For this data, cinnamaldehyde, which is a cinnamon flavoring chemical. So we took human peripheral blood and I isolated the neutrophils and then exposed them to different doses of cinnamaldehyde. And I measured how well they performed one of their important functions, which is oxidative burst. And this oxidative burst helps to clear pathogens by making superoxide, which is a really reactive, toxic oxygen species. And what we found was that at increasing doses of cinnamaldehyde, the cells were not able to perform their oxidative burst as well. And these cells weren't dead, they were just not functioning properly. So if they weren't able to do their oxidative burst, that could mean that they have a decreased ability to defend the body from pathogens. And these doses, what's really challenging about e-cigarette research is we don't necessarily know what doses people are exposing themselves to, but this just shows that it is possible for these flavoring chemicals to impair the function of white blood cells like neutrophils. Another example is something that we're going to focus on when we do the activity later, which is how do flavored e-liquids and specific flavoring chemicals affect the function of immune cells like neutrophils, macrophages, and natural killer cells. And in this study, one of the students before me, who's actually now an assistant professor at UNC, and you'll be hearing from him next week, Dr. Phil Clapp, took macrophages, neutrophils, and natural killer cells and exposed them to either PGVG, so just the base liquid chemicals, or flavored e-liquids. And he looked to see what happened to their function and their release of specific signaling proteins. And I'm not going to show you the data from that yet because we'll see it later, but I just wanted to show you that this is kind of the experimental setup that he used. So at this point, I'm going to go ahead and take any questions if you have them. And in a minute, if you all don't have any, or after we answer questions, I will go ahead and introduce Greg.
Thanks, Elise. Any questions for Elise? You feel free to unmute yourself or drop a question in the chat. We have a few minutes for questions.
Elise, I don't have a question, but the information you gave, about our primary cells, those macrophages and neutrophils, that's an added effect I can give to my microbiology students because we cover how they defend us. And so that's just one more piece of information I could give out. Thank you.
Yeah, cool. That's really good to know. Yeah.
Did we already see some of those cells stain in fluorescence last week, maybe?
Yes, you did. Yeah, Alona showed some of the data from Phil's macrophages last week.
Like that some of the processes were very skinny and almost pulled out, like maybe they had detached from the surface or something. I don't know what the interpretation is, but certainly it looked like intracellular, like cytoskeleton might have changed or something.
Yeah, and when the immune cells move around, which they do when they're kind of exposed to bacteria or other bacterial chemicals that might attract them, they kind of extend these filaments as they move and as they reach out for whatever they're trying to capture. Yeah, those are really cool pictures that he took, and they're, I think, a really good way of showing them being frustrated. Okay, I see a question in the chat. Have you done this study with various flavors? I would assume so. Are some worse than others? So I will say for my neutrophil study, yes, I did a handful of different flavoring chemicals, vanilla flavoring, cherry/almond flavoring, banana flavoring, and cinnamon. And I found that a lot of the flavoring chemicals that had the functional group of aldehydes and that were also aromatic, so aromatic aldehydes, they all had a pretty strong effect on neutrophil function. And then as far as the other specific like flavored e-liquids, we're going to see some of that data later and to see which are worse than others.
Yeah, that was a great question to lead into our activity. Any other questions for Elise? And I guess, Greg, if you want to go ahead and get your slides going here, we can start transitioning if there are no other questions.
Oh, I see one more.
Yeah, some more came in.
Oh, Simone asked, or we've got two more. So Grant Story asked, maybe I missed this, but what types of problems are associated with epithelial damage versus immune cell damage? That's a good question. So obviously, when we're thinking about the whole respiratory tract, they're all very interrelated. So when you're inhaling something that's toxic, it's likely that it's probably going to affect all different types of cells. And so we're really interested in all of them. In certain diseases, you'll see different cell types responding in different ways. Like in COPD, that's often a lot of times associated with overactivated neutrophils and macrophages that aren't repairing things properly. But if your epithelial cells are damaged, then that can also, inhibit your ability to breathe well, or the barrier between the air and your body can be disrupted. So it's really all of them working together that are usually happening in disease states. Let's see, Simone asked, do aldehydes create more superoxides and therefore are more toxic? So there is a kind of complicated answer to that question. So aldehydes can adduct proteins in the cells, they can attach to them. And then when the cell is trying to protect itself from those adducted proteins or to keep proteins from being adducted or undo that, that uses up a lot of the antioxidants in the cell. And so then the cell can be more susceptible to what we call oxidative stress, which is when there's too much damaging oxygen species around. So it's kind of indirect, like the aldehydes themselves aren't making superoxide, but they can cause stress in the cell that's related to too many reactive oxygen species. I hope that made sense, Simone. Let me know if you want any more clarification. And then Lindsay,
good. Thank you.
You're welcome. Lindsay asked, do you see metaplasia like we would with a smoker? So this is one of those questions that's really hard to answer because e-cigarettes haven't been around for as long as cigarettes have. And most of the e-cigarette users we see have been using them for about 2 to 3 years. And so I don't think, if you had a smoker, I'm not sure you would be seeing that after that short period of time. And at least with cell culture models, they probably can't answer that question very well. We do know that e-cigarettes we think that they have fewer typical cancer-causing chemicals than cigarettes do because you're not combusting something. But that doesn't mean that they can't also have other effects on your airways that aren't cancer, because there are a lot of other airway diseases that aren't cancer. So I haven't seen anything about metaplasia with e-cigarettes, but if anyone else has, feel free to share that. All right, it looks like we don't have any more questions, and we will probably have time at the end too if you all have any more. So at this point, I'm going to go ahead and introduce Dr. Greg Smith, who is a postdoctoral fellow in toxicology and environmental medicine in the lab of Dr. Samir Kalada at UNC. His research combines developing custom inhalation exposure systems and the use of new mouse models to investigate genetic and genomic mechanisms that influence susceptibility to the health effects of air pollutants. I've known Greg now for a few years as both a peer and I'd say a friend, so I'm excited for you all to hear him speak today about rodent models.
All right, thank you, Elise, and hello, everybody, and thank you, Dana, also for inviting me to talk with you today. My slides are going to be about rodent models and respiratory toxicology, in particular, their use for studying the health effects of e-cigarettes. Hopefully that changes the slide. There you go. Okay, so you saw this earlier, this continuum of experimental models that Elise showed. And we're going to move a little bit further to the right on this, closer in biological relevance to humans with rodents, but we also have important considerations as well as pros and cons of using rodent models for this type of research. First and foremost is, as Elise alluded to, is the ethical considerations that we have to make when we use vertebrate animals in research. Every federally funded research institution in the US has to have what's called an IACUC, or an Institutional Animal Care and Use Committee, to ensure that the research that we're doing is ethical and humane. And these committees include researchers, veterinarians, non-scientists, and also members of the public, such as yourselves. And we all have to submit applications to these to the committee to make sure that our research is up to snuff. And so you'll see, basically, for more than 100 years, rodents and usually rats and mice have been used for biomedical research. And this is for a couple of very good reasons. First, they're easy to breed and handle. Certainly rats are very docile. Mice tend to get a little bit bitey, but they breed quickly and they develop very quickly, and into something that has a functional, that's a fully functional organism. So we can really model the interaction between different organ systems and the effects of toxicants on multiple organs in their functional state. And although they don't look it, they're genetically similar to humans. Around 85% of their genes are identical in the same region of the chromosomes as they are to humans. And then one really important thing is that there's wide availability of transgenic mice. So if you want to study a very specific protein, you can actually buy off the shelf, so to speak, a mouse that doesn't have that protein anymore. It's knocked out. And so that's termed a transgenic animal. So you guys might think of like a green fluorescent protein being put in the animal, so then you can track a specific protein as it moves, or a glowing green mouse, basically. And with mice, unlike humans, you have the ability to perform really well-controlled and reproducible experiments, because as we all know, all of our exposures up until the point that we do a human studies experiment are very, very different. We can control a lot of those different exposures in the environment of mice, for example. And so that's one of the benefits of using rodent models. There are, of course, disadvantages. Everything as you go along the line of that continuum becomes more and more expensive. And also what may be obvious is that there's clear anatomical and physiological differences between these rodent models and humans. And then what has historically been sort of a drawback of toxicology studies in rodents, although there's a good reason for it, traditional models that people have used or rodent studies that people have done don't include very much genetic diversity. And as you know, all humans are very different and very genetically diverse. And so that's been one of the drawbacks of using rodent models, or at least the studies that have been done in rodent models to this point. So when I imagine a controlled exposure study, the simplest design that I can think of is what's shown here on the left, where you may have a group of mice exposed to filtered air, a control situation, and then you may have e-cigarette aerosol, for example. And what I want you guys to try to think of, and then hopefully put in the chat box, and if I can see it, is how can we expand this design, just two simple groups, to ask new questions and learn more about the effects of e-cigarette aerosols. So let's see, I'll give you a second to try to type in there before I start listing them off. All right, I see frequency and variable dosing. So that's a good one. Anybody else? No. One that comes to mind for me all the time is diet, whether we can change the diet of mice. I see length of exposure and different flavorings, very good. Different mixtures of chemicals, yeah. That's perfect. Temperature of the aerosol, these are all awesome. Genetic background is a great one. I'm going to talk about that a little bit later. Temperature, yep, the temperature that the mice are exposed, like their body temperature maybe even. Age is a very important one. So older mice may have totally different responses. Sex, males versus females, pregnant mice, awesome, you guys, you got it. Stress, crosstalk, okay, cool. It seems like we know our stuff. So any of those could both complicate and allow this exposure design to be to allow us to learn more things. And so similar to what Elise was talking about earlier, we want some data out of this. And so the data that we look for, let's just use inflammation as an example. So does e-cigarette aerosol cause inflammation? We're going to look for biomarkers of inflammation in different regions of the mouse. And this could be looking for inflammatory chemicals in the blood. We can also analyze the lung lining fluid by lavaging the lungs out with saline. And so you can see whether there are inflammatory cells that accumulate in the lungs. Another thing you might do is collect tissue and do gene expression studies, looking at different changes in proteins or lipids. And then a classic thing to do is a toxicity study. People may actually just look at sections of the organ tissue on a slide and see if there's morphological changes. So that's called histopathology. And you may also have toxicity assays. And then one of the really important things that mouse models or animal models in general allow us to do, as I mentioned, it's a whole organism, so we can do physiological studies. We can actually monitor the breathing changes, for example, that occur after exposure to chemicals in e-cigarette aerosols. And then another thing you might be able to do is do co-exposure or co-infection models, where you would have cohorts of mice exposed to e-cigarette aerosol and then tag that on with a viral infection. And that's something that's relatively easy to do in mice. So we have our experimental design, and we want to get the aerosol to the lungs. And so there's a couple different ways that we think about doing that. And three of them are listed here. So you can do a whole body exposure where the mouse is essentially in a box where filtered air is flowing through and it's being exhausted out. And your aerosol is introduced by some mechanism, and you're exposing lots of mice at a time. One of the advantages of this, like I said, is you can include a number of mice in your study. It's a little bit less stressful for the mice in terms of handling, so you can do longer term studies. typically you use a lot of your test articles. So you have to vaporize a lot or aerosolize a lot of e-cigarette for this. Nose only is the next type of exposure. And this is a little bit more directed. So the mice are attached to an inhalation tower, as we call it. And the stream of aerosol is directed to their nose. So they're held in a tube, which allows you to limit the amount of aerosol that's getting on their fur and increasing variability in your studies. And it's a little bit more stressful, so you don't want to be doing very long-term studies this way. You can do multiple per day, but usually they're in there for 30 minutes to 45 minutes at a time, being exposed to aerosols. One of the other advantages of this is you can actually do a puff profile. So you can mimic, or at least better mimic, what a human e-cigarette user would be doing. So you can see that the aerosol concentration increases and then decreases, and you can do that, and you can mimic that in this setup. And then the last one is installation, and so this is where you actually just pipette or inject a bit of liquid with your compound or your flavoring chemical, for example, into the trachea or into the nares, the nose of the mouse. This is really great if the thing that you're trying to aerosolize is really dangerous to aerosolize for you or for the mice in general. Or if it's a very valuable compound that you're trying to introduce. So if it's a drug compound, for example, you want to use a tiny little bit of it and get it right to where it needs to go. And so that's what installation is good for. So in the next slide, I have another question for you guys based on what I just said. So you want to model a 30-minute vaping session using a Joule device, and you want to include that intermittent puff profile I was talking about, and only do it on 4 to 5 mice. So which exposure method would you choose? If you want to pop that in the chat box. Excellent. Cool. I'm seeing lots of nose only. And that for this scenario, that's also what we would choose. And I'm going to show you an example of what our lab looks like as an example. So I have a video in the next slide. Hopefully it'll play for you. So this is going to be a little bit of a tour of our Inhalation Exposure Lab. And you'll see on the right as we go in the door, our ozone Inhalation Exposure Lab, which is another air pollutant. And that is for whole body. And then to the left is what I'm developing for nose-only exposures for mice. Exposure to e-cigarette aerosols. So it kind of jumps. I'm not very good at editing. This is my ozone inhalation chamber that I built. It's all computer controlled. And then this is the aerosol control. So it's the airflow and the vacuum. And so you can see lots of like flow meters and different pressure meters. And this is the tower that I was talking about. And the little tubes that the mice go in are attached to the tower there. On the top is where we actually have a little adapter that we custom made for the Joule. And the system pulls a vacuum on the Joule and pulls the aerosol down into the chamber, into the center column of the chamber. And then the mice are exposed nose only to that. And it's pretty easy to load the mice. They're actually acclimated to these tubes ahead of time for a week or two. So you put the tube in their cage and you introduce them to the tube, and that gets them a little bit more comfortable with the exposure. And so then this last thing here is the control unit that I built. And it's essentially as simple as loading the mice, getting them acclimated, and turning the switch on. And then we program a puff profile into this little microcontroller, which a lot of you probably are familiar with. It's an Arduino. And it controls a relay, which controls the vacuum system. And so that is our exposure lab. And so I'm going to change gears here a little bit and talk about what we're going to try to do with this exposure lab and our new e-cigarette exposure system. And as I mentioned earlier, typically toxicology studies, these types of studies are done in genetically identical strains of mice. And as you know, humans are very different, and we want to try to understand what are those genetic factors that make us all different and why we respond differently to inhaled toxicants like potentially flavoring chemicals in e-cigarettes. And so I have the strains that we use often here as a black 6 and the Fisher rat. They're inbred. They're all genetically identical when we use them. And so how can we model the influence of genetic variation on toxicity of e-cigarettes? And the way to do that is include more strains in your study or outbreed the mice. And so this is an example of a study that was done at NIEHS, which is the National Institutes of Environmental Health Science, by Alison Harrell. And so this is showing a study in a bunch of different mice of acetaminophen toxicity or Tylenol. And so you can see the dose of acetaminophen increasing along the x-axis here, and the toxicity of it increasing on the y-axis. And all of the different strains are labeled here, and each of these lines represents a different strain and its toxic response. And so you can see right away is that there's some strains that respond very early and are very sensitive to acetaminophen, and then those that barely respond at all on the right. And so what we want to do in our lab is try to use this strategy of using diverse mice for e-cigarette research. And so you can imagine that the hypothesis here is that if you treat different strains or expose different strains to e-cigarette aerosol, more strains than just 2 in most of the time, they're all going to have very different inflammatory responses to exposure to e-cigarettes. And then because we can sequence the genomes of these mice, and we know what their the variability looks like, we can kind of, using some fancy genetics, trace back to the actual genes and the nucleotides that are different between these mice that underlie these different inflammatory responses. For example, in strain 2 here, this change in the genome of strain 2 leads to more gene expression in maybe a macrophage, for example. And that leads to that macrophage being more sensitive or more highly respondent to e-cigarette aerosols. And so hopefully what we do here with these diverse mice is better model the human population. And then, of course, when we identify a gene that underlies these increased susceptibility in strain 2, for example, we would go back into the human population in human studies, and we have a much better chance of finding something that actually affects human susceptibility, rather than taking a look at millions of humans. And you can do that much more easily with a mouse model like this. So just to summarize what I was talking about, the kind of key points that I want you to take home. Rodent models, they're commonly used for respiratory toxicology research, and for a lot of questions, they're the best model to use. You can ask questions like we talked about before, with different diets, different sexes, different exposure regimens, and you can even do drug treatments. But these types of exposure experiments in rodents require careful design as well as controls. And the new things that we're trying to add now are genetically diverse mouse populations, which we think are better models for studying the contribution of genes to the adverse respiratory health effects of e-cigarettes. And so that's the end of my talk. And I will try to look at questions.
Thank you. One question is probably going to be, can teachers show that video to their students?
Yeah.
Okay.
Yeah, that's perfectly fine. Yeah, I didn't include any videos of mice or anything.
Right. Yeah. So we will, we are recording this, and we'll at some point, we're also going to chop this up into smaller videos too. But I always thought a neat activity would be to have your students design an experiment and then show them what researchers are doing, as a way of being like, you just thought about an experiment the way researchers are, as kind of a confidence booster. But do we have any questions?
Yeah, that would be awesome.
Yeah.
Yeah, I have a question. My name is Grant Story. I'm a teacher at Cleveland STEM High School out in Seattle. So my question, I thought that's really fascinating, especially the animal models and the different deliveries. But my question was around maybe like an addiction model. Do mice choose to vape like a human would choose to vape? And is there like a model or an experimental model system for that type of behavior?
That's a great question. Both mice and rats are typically used for addiction models. Mice are a little bit harder to work with, at least I've heard. But rats for sure are used for addiction research. I don't know the logistics of self-administering a e-cigarette aerosol, but rats would, if they were addicted to nicotine in the e-liquid, they would self-administer it. And so you could look at questions like that, whether certain e-cigarettes are more, formulations are more addictive than others, for example. But you can do that with these types of animal models. That's a great question, Grant. Thank you.
Yeah, thank you for your answer.
Let's see, you have a few other questions. Do you use bronchodilators on any of the mice prior to E-Cigs?
We have not yet, but we certainly could. So we could inject those and see how that affects the delivery of the e-cigarettes. One of the questions that we have all the time is whether when you expose mice in any of these ways, does it actually get to the lower airways? And so one of the ways to increase delivery is to open the airways, and you could do that with a bronchodilator or have them exercise for sure. But we haven't done that yet.
And then another question came in, do you have any data on long-term exposure to the aerosols?
I don't yet, but there are a number of studies going out to several months in mouse models, I believe that it was, and there are definite changes over long-term exposure, not necessarily as you would expect, like very strong toxic responses, but there are changes in the macrophages in there, like Elise was talking about. Mice that are exposed to e-cigarette aerosols for a very long time have these, their macrophages change their phenotype and they start to look very different. And that may belie or underlie some of the changes that you see in macrophage function with e-cigarette aerosol exposure. What we call them foamy macrophages. They start to get these like lipid droplets in them, and so they look very different. That's the one study that I'm aware of long-term exposure to e-cigarette aerosols. Thank you.
And in that study, they also found that the mice who were exposed to e-cigarette aerosol were not able to respond very well to a flu infection. And we'll be hearing more about e-cigarettes and viral responses in the last week of the series.
Great, and a couple people commented on, how it's neat for students just to see the creativity that goes into designing these types of experiments. And someone else Julia asked about how long did it take to configure the Arduino interface. I know some students would be interested in seeing that you all used Arduino.
Yes. Yeah, it didn't take me as long because I actually had some help from a friend who had built a similar device before, but there's a lot of resources online, and I think that this is what a lot of students probably are doing too, is there's tons of resources for Arduino. And basically, if you think of a process that you want to do, just like turning a switch on and off, that can be done with the Arduino. And so I was able to find some code that other people had already posted online and use that, adapt that for turning on and off the relay that controls the e-cigarette aerosol.
Cool. There are a couple other questions coming in, and we're going to need to turn it over to Elise soon. I will say Simone asked about specific research papers, and we will certainly provide links to some, and you can also look up papers coming out of the Jaspers lab and the Collada lab at UNC. So we can definitely help with that. And then, Greg, you'll see some other stories or questions came in about mice being sacrificed and how comparable are they in developmental stage to an adolescent human. I was wondering if maybe you could just answer those in the chat so that we could move on to Elise. But I really appreciate you being here and letting the teachers know about the work y'all are doing.
All right, yeah, thanks for inviting me. I'll try to pose some answers in here.
Yeah, that would be great.
Oh, and you want to stop sharing?
Sharing. Yep.
Yeah. All right, we're moving on to our last micro talk, moving up the experimental model chain to talk about human subjects research in respiratory toxicology. So back to this experimental model spectrum, now we are going to talk about humans, which are the most biologically relevant of anything we could study. And I guess they're not really a model because humans are what we are interested in studying, but they are probably one of the more challenging experiments to do. So let's talk about a little bit about human subjects research, which is any systematic scientific investigation that can be either interventional, so you give people a treatment or have them change their behavior, or observational, you basically just bring people in and observe them as they are. And it involves human beings as research subjects, commonly known as test subjects or participants. Some people like to call them volunteers, although a lot of times they are paid. I'll refer to them probably as all of these things at some point during the talk. But similar to animals, human subjects research also has to be approved by an institutional review board. And that's to ensure that the studies are ethical, that they minimize risk and maximize benefit to the study participants. Because there have been a lot of historic examples of people being exploited to do human subjects research, treated very poorly. And so this type of research now is very highly regulated and has to be approved by this institutional review board before it can be done, in the hopes that protects people, but still allows us to do human subjects research that can be really beneficial to human health overall. There are, like with any model, there are advantages and disadvantages of research with human subjects. And like I already said, the major advantage of human subjects research is that it's highly biologically relevant. It's what we really care about as toxicologists, which is human health. And you are able to take samples from different demographic subsets. So you can take from males and females, you can take from people who have a disease or who don't have a disease, or people who are using different types of exposures. So it does allow some flexibility in what population you're studying. There are a lot of disadvantages and challenges to human subjects research. You're not able to explore molecular mechanisms as well, because you're not able to knock out specific proteins in humans like you can with mice. And you can't just expose people to any chemical that you want at a bunch of different doses. It can be really time consuming and costly to recruit all of the people you need, and costly because it requires a lot of resources. You need to have clinical staff, clinical study coordinators who are able to handle bringing people in and taking samples from them. So you usually need a clinical space. It requires a lot of paperwork and applications. You have to be able to protect private health information. So there's a certain level of infrastructure that you need to be able to do a human subject study. And like Greg talked about, there's a lot of variability between people, both in genetics and in what they're exposed to all the time. And so that variability can mean that it's really hard to control what people are being exposed to, especially with e-cigarettes. When we bring people in and take samples, a lot of times they have to fill out a questionnaire. But in the end, like, they don't always even know what they're exposing themselves to. Like, one time someone told me they were vaping 300 milliliters of e-liquid a day, which obviously is not, they're not exposing themselves to that much. So that variability and the differences in exposures can be really challenging, and it can mean that you might not be able to detect the effect that you're looking for, or that you might need a lot of people to overcome that variability. So how do we conduct a human subject study? It's a pretty long process. It starts out, the researchers conceptualize the study they want to do, and then they have to submit an application to the IRB, which is reviewed. Sometimes they ask for changes or clarifications, and eventually it's approved. And once it's approved, you can start recruiting subjects. And a lot of times this is advertised on something like clinicaltrials.gov or on the university's clinical research page. And then when we find out that someone's interested, the clinical study coordinators screen them over the phone to make sure that they fit the eligibility for whatever study we're doing. And then the subject comes in for consent, where they go through the whole protocol and basically say that they understand the risks that they're taking and sample collection. And sometimes people might have to come back 2 or 3 times for different exposures or samples, depending on the study. After we get the samples from the people, sometimes we'll do assays with the live cells or with the samples. Most of the time, though, we process those samples and store them in the freezer until we have samples from all of the people who are coming in. And so this recruitment all the way to sample collection is repeated for every person that needs to come in. And once we have that whole cohort of people, then we can analyze the samples. So this can often take years to complete, even just the sample collection process, and then you get to analyze the data. So it can take a long time to get results. The cells that we are interested from human subjects, and that a lot of other people studying respiratory toxicology are interested in, are nasal epithelial cells and bronchial epithelial cells, like we talked about in the cell culture talk. So when we talked about the primary cells, to get those cells, it's considered human subjects research, so we have to go through this whole process as well. And there they are again with their cilia and then looking nice and polarized. We can get immune cells from the lower airways as well. So we collect induced sputum, which is where the subjects inhale a nebulized saline solution, and that causes the airway secretions to be released, and people cough it up and out, and we can take that, and there are cells in that we can study, such as it's mostly macrophages, but there are neutrophils as well, and in diseased people, there's sometimes a lot of neutrophils in those bronchoalveolar lavage macrophages, which we get through bronchoscopies, and that's what they look like. And then again, with the blood, we can get neutrophils, monocytes, natural killer cells. So we can take all of these samples from people who come in, and we can take them from different populations of people. We can take other samples and data too that are really useful for our studies, like cell-free fluids, where people might wash out their nose, or that cell-free component of sputum or the lower airways. We also collect blood serum and urine, and those are all used to measure important levels of proteins. For e-cigarette research, we're usually interested in serum or urine cotinine, which tells us how much nicotine someone is consuming, because it's a metabolite of nicotine. But you can also look for other biomarkers in these fluids as well. They usually fill out smoking and vaping diaries, and that's to give us a sense of what the subjects are exposing themselves to. And they're not always the most accurate, but they can still be useful to determine what flavors people are using or what devices people are using. And it's really important to get these to make sure that the people are assigned to the correct group, and that you don't have someone who's smoking a lot in the vaping group or vice versa. Usually we don't include mixed users of these because we want to be able to separate out the effects. We can do spirometry on these people, which measures lung function. And so this is what it looks like when someone does it. Your nose is clamped and you have to breathe in and out of this tube, and there's a machine that measures the volume that you're breathing and also the speed at which you're exhaling. And this clinical coordinator leads the people through a specific breathing routine to take the measurements that we want. And then we've also done Centigraphy, which measures particle clearance from the lungs using radio-labeled particles and a special camera. So people will inhale these radio-labeled particles, and then they sit in front of a camera, and we can watch those particles get cleared out of their lungs. So this is what the image looks like. You can see these are the green particles, the central and the peripheral part of the lung lobe. And over time, you can see these particles cleared out. And if someone isn't clearing particles very well, you will also see that. And you'll hear a bit more about that next week when Phil Clapp presents. So the overall experimental design for e-cigarette research with human subjects is that we would bring in non-vapors and vapors. Sometimes we also include smokers in this set. For some projects, we might have asthmatics or non-asthmatics. We can take from a lot of different populations. We collect all of the samples that we want, and eventually we will analyze and compare those samples for lots of different endpoints. And some of these endpoints can be very similar to what we're measuring in our cell culture experiments. And sometimes we'll do that to see if what we're seeing in our cell culture when we expose the cells to e-cigarette aerosol is the same as what we see in people who are exposing themselves. And that can be a really nice way to link the two different types of research. Some examples of this type of research. This is one of my projects about whether e-cigarette and cigarette use impacts the nasal microbiome. A lot of people have only really thought about the microbiome in the context of the gut microbiome, but anywhere that touches the outside world, you have a microbiome. So on your skin, your nose, for example. So we collected nasal samples from smokers, non-smokers, and e-cigarette users, and we analyzed this using high-throughput sequencing of a gene that's specific to bacteria and can be used to identify different bacteria. And we were interested in this because the microbes in your nose are an important part of your host immune system, and you are interfacing with them all the time, but no one knows whether they're perturbed at all by e-cigarette use. Interestingly, we didn't find any differences in bacterial diversity or like the number of bacteria or how many different species are present in non-smokers, e-cigarette users, and smokers, but we did find a pretty significant difference between males and females, which was in some ways surprising because it hasn't been reported that much, but we have seen other sex differences in respiratory immune responses. So this was a really cool finding. And then we were able to adjust our data for that difference in baseline. And what we saw was that there were unique communities of bacteria in e-cigarette users, smokers, and non-smokers. So this graph here, each dot is a person, and you can see that the different groups cluster differently, and there are specific bacteria that are driving that clustering that we can determine using computational analyses. We have also studied different immune phenotypes in the airways of people who use different types of e-cigarettes. So we've been talking a lot about Juul and how that's become so popular. And Bob talked about the nicotine salts in Juul, which makes it a bit of a different exposure than previous generation devices. It also aerosolizes the e-liquid slightly differently. So for this study, we took induced sputum from a bunch of different human subjects, and we looked for, the cells in the sputum, and we looked for inflammatory markers. And we found that Juul users had a lot more sputum, and they had a lot more cells in their sputum. And those cells were mostly neutrophils, indicating there's some sort of inflammation going on. But we didn't necessarily see any increase in the markers of inflammation that we happened to look for. And so we're still kind of trying to figure out what's going on in these subjects. In these cells, we also measured mitochondrial membrane potential in their macrophages, because this can be a marker of how inflamed the macrophages are, what type of phenotype they have. And we found that in the vapors and the Juul users, the mitochondrial membrane potential was increased. And this is something that macrophages do when they switch towards a more inflammatory phenotype. They start to use their mitochondria to produce reactive oxygen species instead of using it for normal cellular energy purposes. And so this is an ongoing study that we're still trying to understand these differences in Juul users, but we definitely see some pretty pronounced differences in comparison with other e-cigarette users, smokers, and non-smokers. So with that, I'm gonna go ahead and end that talk, and I'll take any questions you all have. And then after the questions, Dana's gonna transition us into doing the activity together.
Yeah, why don't you go ahead and stop sharing and I'll switch over.
Okay.
Thanks.
Yeah.
Yeah, feel free to ask questions, pop them in the chat. Victoria, do you mind monitoring the chat for questions?
Sure, yeah. So let's see here. So I'm gonna go back up. I think we.
I see a question in the chat from Liana. Do human research institutional review boards require testing of a range of socioeconomic or ethnic groups for research that is intended to be applied to a broad range of patients/subjects? Yes, that is something that they look at, and typically, at least what we use in our applications, is that we want our cohort to represent the same demographics that North Carolina represents overall, or that is local to our area, because obviously if we don't, depending on what the breakdown of people near where the research study is happening, it can be hard to recruit people that aren't, that if you're looking for a lot of a specific demographic, but they don't live nearby, it can be really hard to recruit that. And I will say that there have, not in the specific field, but there are researchers who are specifically focusing on specific socioeconomic or demographic groups' responses to toxicants or likelihood of having certain disease states. But that's definitely something that we include, and we look for in all of our researches to see if there are any impacts of that. Although we don't really focus too much on socioeconomic status, just because it's not really it's not really impacting the biological state of the people who are coming in, but in some studies they do. Do any of these studies look at THC liquids? Yes, so no, are not allowed to do any controlled studies using THC because it is a Schedule I drug. We do ask in our questionnaires whether or not people are regular marijuana users. So we have that as a column in our data matrix that we can look to see if there are any effects that are driven by that. And they're allowed to tell us that information because this is private health information. It's protected. So it's not like it could be used to incriminate them. And we usually find that about half of our e-cigarette users are also regular marijuana users. But we aren't able to do any cell culture studies where we expose them to THC, unfortunately. Are your subjects vaping normally, and how does that impact what you're looking at? Can you control those extra variables? Yeah, so when we collect the smoking and vaping diaries, we usually ask them to report either how many puffs they're taking per day, how many pods they're using, or how many milliliters of e-liquid they're using. And they usually do those diaries for 2 to 3 weeks. But again, they're not necessarily the most accurate reports. So there's only so much that we can control for. And we do the best we can, but again, it's not always possible to control all of those extra variables. Measuring the serum cotinine is a way to approximate how much they're using because it can, it indicates how much nicotine they're using. So most of the Juul users, they're all using a 5% cool mint or menthol flavored Juul. So all things equal, you would expect that nicotine amount to maybe reflect how much they're using. But again, how much different people are able to inhale and how much gets into their bloodstream might vary from person to person. And Craig asked, I'm assuming your subjects using a wide variety of flavors. How does this affect your results? So in the previous generation e-cigarette user group, there are a wider variety of flavors, but in general, for the Juul user group, it's pretty much all mint. So that's actually pretty consistent, but it can affect the results. I haven't seen so far it affecting the results, but again, I don't think we have enough people using every single flavoring to have enough power to detect that. But it's definitely something that I would like to do if I had an infinite number of subjects and a lot of time and money. And you'll be hearing some about humans being exposed to the cinnamon flavored aerosol next week with Phil. All right, I think we're
Thanks,
about done.
Elise.
Yeah, and I'll continue to answer any in the chat if you all think of any.
All right. So we're and thanks, Greg, if you're still here.
I am, you're welcome.
Great, I appreciate both of you having such informative presentations and lots of good questions, and hopefully you all are seeing lots of possible connections. And I think I've shared this with some of the teachers who were on the webinar before, but I used to teach biology at a community college, and I always tried to pull in health as much as I could to make the biology relevant, because I had a lot of non-majors who didn't understand, why they had to take biology. So I think e-cigarettes and what scientists are doing to study their health effects can just be really kind of sticky in a sense, and help students understand, how to apply biology to our health, and maybe attract them into careers too. So hopefully this is useful. So we're going to transition now into our lesson, and we are starting a little bit later than I had hoped, which is fine. But I think what we're going to do is I'm going to change it up a little bit, and we're not going to go into breakout rooms, but we're going to instead kind of go through this activity together and make it, interactive with Elise and I. So I will go ahead and place into the chat though. It's the link to a Google Drive folder, which in this folder is a PDF of our lesson, as well as a companion slide set to go along with the lesson, as well as the student worksheet. And we'll talk about all this, but you have all these things. This was the first lesson that Elise and I wrote a couple of years ago. We had teachers review it. We piloted it in classrooms. We were kind of targeting AP biology when we designed the lesson. But one thing that we like to do is get data into the hands of students so that they can kind of discover, rather than you telling them about e-cigarettes and health effects, they kind of discover what those are. So this lesson is called Exploring the Health Effects of Flavored E-Cigarettes on the Respiratory System. And our guest presenters, Elise and Greg, have done a nice job of kind of giving you some background. But this is just an overview, and Elise introduced this earlier. So the lesson that we're about to move through is where researchers from Elise's lab looked at macrophages, neutrophils, and natural killer cells, and they exposed them to either the base liquid, that PG-VG mixture, or flavorings, and then they wanted to look at the effects of these. And so that's what we're going to be looking at in this activity, is data from these studies. Elise showed this earlier. This is kind of their experimental design. So they use these cell culture plates. And then one thing we have included in the lesson is a little bit on statistics so that students understand what the asterisks mean. Let me just move my bar here. Elise, do you want to do this slide?
Yeah, so we know that some students may not necessarily, know statistics or have taken it or be comfortable interpreting these graphs, because these are the graphs pretty much straight from the paper. And so this slide is just going over that p-values indicate when a comparison is statistically significantly different, which is important because it tells us as researchers that our results are likely real effects of a treatment or an experiment versus being random effects due to variability in the experiment. And the number of stars just indicates how significant it is. It doesn't necessarily mean that they're the most different, but it means that they're most likely to be truly different from each other in real life. So we have a couple of quick questions here. Do you want to do these, Dana, or do you want to just move to the next slide?
We can just we go through them real quickly and maybe not wait on answers, but just.
Okay. So the first, and you could ask this of your students to put it in a chat or to answer it live or something. The first one is, which graphs show statistical significance? And that would be graphs 2 and 3, because you see that those have the bars and the asterisks. And then which graph shows that A and B are the most likely to be truly different from each other? And that would be graph 3, because you can see that it's more statistically significant. And the next slide. But it's also, I think, important to think about biological versus statistical significance. I like this quote: the conclusion that there's a statistically significant difference indicates only that it's unlikely to have occurred by chance. It doesn't mean that it's necessarily large, important, or significant in the common meaning of the word, as in like significant for health. So just because the change is statistically significant doesn't always mean that it's significant to the overall function of the cell or the organism. And we can see these changes and report them when we publish papers, but it's sometimes really hard to know what amount of change is important biologically. And this goes back to, using different model systems and not knowing exactly what people are being exposed to in real life. But it's just kind of a caveat of scientific research. And sometimes a change is not statistically significant, but it may be really important biologically. So let's say we observe, maybe a 50% decrease in something in e-cigarette users, but because we're using human subjects, the variability is really wide, and therefore it's not statistically significant. So these are kind of tricky things to think about, but I think it's a good mental exercise just to remind yourself that they're not equal and that you have to consider that. And it's something for your students to think about whenever they read research or read an article, to think about what the different meanings of those two things are.
Yeah, thank you. So Elise and I designed an activity. I'm going to actually move to this slide first, that we were thinking teachers could kind of use a jigsaw approach for. And this could, where you could do all of these or pick the ones that are most relevant to what you teach. But we took 6 different data sets, and you can see little screenshots of them here. But in person, when we did this, we would hand small groups one data set along with some guiding questions to guide the analysis, and the students would input, their answers onto a worksheet, and they would also prepare summaries, and they could report out to their class. So each group would kind of teach the others. So we're going to go through some of these tonight, and these are the 6 guiding questions for the activity. So the first one looks at PG-VG and how does it impact phagocytosis and macrophages and neutrophils. And I know some of you have to teach about phagocytosis, so this would be a way to maybe reinforce that content. And then some of the questions look at certain flavors. Some of you asked about flavors earlier, so we'll look at some data on different flavors, how that affects macrophages and neutrophils in terms of their phagocytosis, also cytokine secretion, which is important in inflammation. And then we're also looking at something here called neutrophil extracellular traps, which we'll tell you about in just a minute. And that might be new for some of you, because I think that was discovered about 10 years ago. So again, these are guiding questions. So each group could get a guiding question. And again, as a teacher, you might say, I only want to do questions 2 and 3. So that's where you could pick and choose what you want to do. These are just screenshots. And this is just a reminder that we have the guiding questions, which is that center screenshot here. And then there's a worksheet that goes along with this too. And these are available. The guiding questions are part of a PowerPoint, and the worksheet is a Word document that can easily be, converted to a Google document. And we're happy to support you all. As if you want to do this activity in some form, let us know. We of course designed this lesson for in-person use. We think it's highly adaptable for an online environment, but we're happy to support you if there are things you need to facilitate online teaching. Let us know. So I think we're just going to go through a couple of these together as a group. I'm just going to give you a moment to kind of digest this slide, and then Elise and I will kind of debrief it with you all. So here, this is guiding question 1, do the components of the e-liquid base. So this is only a nicotine-free e-liquid, no flavorings. Does it affect phagocytosis in macrophages and neutrophils? And hopefully most of you know what that is, but that is when these, the cells kind of ingest bacteria or other pathogens. So it's a lot of times people think of them as like little Pac-Mans, right? So that's the process that we are looking at here. So take a moment and just look at this slide.
And I see a question real quick in the chat from Suzanne. What, if anything, did students learn about the topic before looking at these? So they basically got the talk that I gave last week, the intro talk. They essentially got that talk plus maybe half of what I did for the cell talk and the biologic and the significance slides. And I think those slides should also be available in that Google Drive folder if you want to see what intro slides we had given them. And you would be welcome to include any of the ones that you have learned or seen as part of this series.
Yeah, that's a good question. And that really is going to depend on, what you teach. Some teachers, when I taught biology, we talked about phagocytosis when we talked about endo and exocytosis, for instance. But we do have introductory slides that go with this lesson as well. So you can see here that we have different concentrations of the base liquid from 0.25% to 1%. We have 2 different cell types. And then they challenge these cells with the Staphylococcus bacteria, and they measure phagocytosis after 4 hours. So does someone want to unmute themselves and tell us what you're seeing here? Julia, do you want to unmute yourself, or am I putting you on the spot?
Well, it looks to me like macrophages were affected less than neutrophils based on this being kind of similar, but your y-axis are different.
Mm-hmm.
So the 100 level on your macrophages is lower than your 100 on the neutrophils. So you have a decrease of going in your macrophages from about 100 down to 90. And on your neutrophils, you have a 100 down to about a 62. So it appears the neutrophils are more affected by this e-liquid base, but I am not smart enough to remember what your little lines above the bar graphs mean.
Oh no, you definitely got it. It's both significant. They're the same amount of significance, and that's probably because you can see that the error bar on the neutrophil graph is a lot bigger than the error bar on the macrophage graph. So even though this goes back to that biologically versus statistically significant concept. So you can see that the difference is obviously a lot more neutrophils, but they were a lot more variable in their responses.
Thank you, Susan.
I see question. So is the bar bracket supposed to compare only the highest? So the bar, it represents the standard error. That's maybe something we should add to the yellow box down here. But the mean is how high the bar goes up, and then the standard error is the little bracket on top of it, and that can be used to eyeball statistical significance. So if if you envision the bracket going above and below the top of the bar, if the brackets don't overlap, that's kind of a signal that they're probably significantly different.
I know that's something they cover in AP biology, so that was one reason why we included error bars in the lesson as well.
Was a post hoc test run to determine which concentrations were different from each other? I would have to go back to the paper to look at the specific statistics run. Typically, yes, we do, but based on how these look, I don't know that I would expect any of those to be statistically different, at least from the different doses. But to have a dose response officially, the doses do have to be different from one another in addition to being different than 0.
Cool, so we'll move on. I think that last one was just interesting because that was just plain base liquid, and we're learning more and more that base liquid itself is not a benign substance. So that's something to keep in mind because it's, again, those things are safe for consumption, right? Generally recognized as safe, but that's in terms of consumption and not inhalation. So that's something to keep in mind that connects to content we learned last week. So here's slide number 2, and this is one I think y'all will be interested in based on your questions. So this is looking at flavored e-liquid, so nicotine free, but now different flavors. And you can see the PGVG base liquid is now the control on the far left. So these graphs, the top is looking at macrophages, bottom neutrophils, still looking at phagocytosis. So similar experimental setup, it's just now we're looking at the different flavorings. So I'll let you look at this for a moment.
And if anyone has any ideas in the chat why we might have used these dilutions or used any dilutions at all of the e-liquids, that's a question we asked the students. So I'd be interested to see if you all have any ideas why the dilutions were used.
Yeah, make sure you might have a little zoom box covering that, but in the upper right-hand corner, you can see the different dilutions of e-liquid. So does anyone want to unmute and tell us what you're getting from this analysis? Which flavors you might want to avoid?
I'll go ahead and take a shot at this.
Thank you.
Definitely looks like the cinnamon. It's affecting the macrophages and seems to be the main one affecting the macrophages, but it also affects the neutrophils quite a bit. For the neutrophils, it looks like the hot, is it cinnamon candles? And the banana
cinnamon candies.
Yeah, candies. The banana pudding and banana also are having quite a bit of effect on the neutrophils. The menthol has somewhat of an effect compared to the PGVG control. So it's much more widespread, the effects in the neutrophils than the macrophages, which is interesting.
Yeah, and we put some material in the lesson because it does appear that neutrophils are more sensitive to the flavors, and the scientists don't know exactly why. There are some different hypotheses, and we present those in the lesson, but that could be something you could have your students do, is go learn more about neutrophils and think about why they might be more sensitive to their environment. Elise, do you want to add anything?
Yeah, so as far as the dilutions, yeah, I think some of you are pretty close in the chat. Talking about that's how much people are taking in that actually reach the cells. And again, we still don't actually know exactly what doses people are exposed to in their body, but the idea is with diluting the e-liquid that we're going to get closer to a more relevant dose inside a person, because obviously when you take a liquid and you aerosolize it, it becomes a lot less dense. So it's not like people are like dumping e-liquid down their lungs. And so if we didn't dilute the e-liquid enough, then the cells would basically just die and we wouldn't be observing effect of the chemical on cells that are functional. And that's actually what you see a little bit in the sinucide group with the neutrophils. That increasing phagocytosis is actually because the cells were dying and it kind of interfered with the assay and with the way that it was being read out. And then I see also questions about what's the difference between menthol and menthol tobacco, and another one about off-brand. So these were just a mixture of e-liquids. All these e-liquids were purchased because they were really popular at the vape shop. So it is a mixture of brands, and sometimes like the different menthol flavors can taste different depending on how much flavoring chemical is in it. And the menthol tobacco may have a little bit more tobacco flavor than just the menthol. And then another question, do the flavors that are having an effect, is there a chemical similarity like they are aldehydes? Well, you're actually spot on there. So the cinnicides, so cinnamaldehyde actually has driven a lot of these responses, which is a pattern you might see as we go through the lesson. And then is it just tobacco flavoring? Is there actually tobacco? There's not actual tobacco in it. Tobacco flavored E-cigs are actually relatively unflavored because it's just the nicotine in it itself and kind of the aerosolization products that sort of give it a tobacco-like taste, but there aren't a ton of flavoring chemicals in tobacco. Sorry, one more question, then we'll move on to the next slide. But how did they get greater than 100% phagocytosis? So this, maybe that's phagocytosing yourself, that's funny. So when we do these experiments, it's usually within each person, we would compare the phagocytosis with a specific flavoring chemical back to the PGVG. And if the chemical doesn't have a lot of effects, there might just be some variability in the experiment that means that well phagocytosed a little more than the other well. And so you can, it's the relative phagocytosis compared to the PGVG. So you can have more than 100 if that well happens to be a little more phagocytic for whatever reason. It's just part of the variability of the assay.
And I think we'll move on a little bit. We're not going to cover all of these. This is a similar graph. I'm going to skip it just because I think you can interpret in a similar way than as to the last slide. But I wanted to just introduce these neutrophil extracellular traps because I think this could be an interesting thing for some of you to teach about. There's a lot to digest here, and maybe Elise, it might be quicker if you don't mind just orienting them to it really quickly.
Yeah, so neutrophils release DNA and other molecules outside of the cell as part of the neutrophil extracellular trap, and DNA is a pretty sticky molecule, and so that's why it's able to attach things to the DNA. You can visualize this formation using fluorescent microscopy. So in these images, you can see the neutrophil DNA is stained blue, and usually we included DNA dyed just so we can see where the cells are. And then neutrophil elastase is stained green so that you can see the extracellular traps being formed. And those are these kind of sticky outy projections with the red arrows. And the way that this experiment was set up is that the cells were exposed to PGVG, cola, or hot cinnamon candies, or cinnacide, that's on the left column. And then they were monitored for 1, 2, 3, and 4 hours after they were stimulated to make their traps. So they don't just like make them for fun, they have to be stimulated to do so. So we exposed them to the flavoring chemical, then stimulated them, and then watched to see what would happen. And what you can see is that if you look at the PGVG control, it takes about 4 hours for the extracellular traps to start forming, and that's what we would expect. And at the 1 and 2 hour mark, you can see that they're just kind of hanging out in their cells, and there's some of that protein in the cells, but it's not actively kind of coming out of the cells. With cola, you can see that there is maybe a little bit more trap formation. It kind of looks similar to the 4 hours, though, maybe a little more activation at 3. But what gets really interesting is with the hot cinnamon candies, we don't see any extracellular trap formation at all, even after 4 hours. And with the cinnacide, we see it starting at 1 hour, which is actually the cells basically lysing and dying because that was so toxic to them, which we didn't know at the time, but that's kind of why that happened so early. And this is a graph that you can definitely break up. You could just take away everything except 4 hours, or take away everything except 2 of the rows to simplify it for your students. But let us know if that makes sense, what you all think of this.
Yeah, and you can also, this is also another opportunity to, to introduce or reinforce qualitative data. And something else that teachers have said they're interested in here when they learn about these, the cell is extruding its DNA, which is a very expensive molecule biologically to make. So you can have your students go learn about this process and why do cells do this, and they'll discover that I think this was discovered about 10 years ago. So it could be neat for those of you who get into the immune system a little bit more. It could be neat to teach about this. And real quickly, I'm just going to show you the next slide because this
I'll
is
answer the questions in the chat.
okay. Yeah, this is basically taking that qualitative data that Elise just presented, and this is the quantitative data behind it. So that's a nice parallel to have your students look at that. So you can see here, again, the Sinicide was really causing problems in terms of, you saw a lot more, the DNA concentration goes up because they're extruding their DNA when they form those traps. So yeah, Elise, if you'll answer the questions, and I'm just gonna advance ahead. We do have discussion questions. We have an answer key for you all. And here's a summary, and then we're gonna take questions, but I know we're right at time. So this would be a kind of a summary. So they go through the data analysis and they see that certain flavors, especially cinnamaldehyde, really decreases the ability of all three of these cell types to do their jobs effectively for the immune system. And then they reflect on what are the consequences of that. And so, yeah, Elise, do you want to answer any questions?
Yes, I'm answering a couple in the chat.
Okay. So I know that was a zoom through, and next week we are going to have more time. We can debrief this at the beginning, and I promise we'll do an activity in breakout rooms. So thanks for bearing with us as we're not teaching online every day like all of you are. So we're still learning about the timing of things, etc. But that's really what we wanted to show you. And you have the link to the materials. Here's our email again. You have the email from me if you want to email me about the lesson specifically. Any questions that you want to ask aloud? And so we know, that y'all all teach different things. You teach different levels of students. So we're hoping that you'll, reflect on what's useful to you. And next week we will have you all kind of share that with each other in groups as well. And so those of you on the call tonight, we had about 27 teachers here, and y'all will be back next week. And it's going to be nice to get to know all of you through these events, but also through just email. So please let us know how we can support you as you think about how you want to integrate content that you're learning into your instruction. Any final questions? I know a lot's happening in the chat.
Do we have any evaluations for them to fill out?
Yes, we do. I'm going to go ahead and drop the evaluation link into the chat if you want to do it right now. And then I'll also send the link out tomorrow morning. We really do appreciate your evaluation because it's going to help us to improve next week's session. But also it's just going to help us better understand how we can, make these materials and these online events and workshops, more relevant to you all. And Elise is going to be taking some of the evaluation data and sharing it at a Society of Toxicology meeting in March. So that's kind of exciting too. All right, well, you're free to go. And if you do have questions, you're welcome to stick around, and Elise and I will stay here for a little while longer, but we look forward to seeing all of you next Thursday at 4:15. And you can expect an email from me tomorrow with follow-up items for today. And we'll post the presentations in that same folder as well. So thanks everybody, have a good night.